Hello, everybody, and welcome to a Aquafix webinar for our aerobic series on microscopy for filament identification in activated sludge. My name is Saylor Gilbert, and I'll be walking you through some things today. At Aquafix, we specialize in creating bioremediation and bioaugmentation products. We also work with aerobic and anaerobic wastewater systems and operators to come up with custom solutions. Generally, when we're talking about microscopy for the purposes of wastewater diagnosis and prevention of upsets, we would include an entire evaluation of floc, filaments, bulk liquid, and higher life forms. All of these things play a role within identifying what sort of issues can be taking place within a system or identifying what sort of things could potentially happen within a system. But for today's webinar, our primary focus is going to be on filament identification and exploring some of those filaments and what sort of issues that they can cause and what sort of ways they manifest within a microscopy sample. So in general, why do filaments matter? They, in and of themselves, can be very problematic by physically causing bulking, foaming, and pore settling. But furthermore, they are indicators of health issues, and they're very strong indicators at that. Filaments will generally only develop and form under specific environmental conditions. And so if we're able to use microscopy to identify those filaments, we can then identify what sort of unique environmental conditions are taking place to cause the growth of that filament. And then we can identify what sort of issues or nutrient imbalances or toxicities might be happening within the system. And we can work towards alleviating those issues and ultimately alleviating the problems within the wastewater plant. The main focus of this webinar is going to be on filament identification, but this can be a strong resource to use to remember how to prepare a slide or learn how to prepare a microscopy slide for wastewater routine analysis. Preparing your slide for microscopy is very straightforward. You're going to need your microscope slides. These are standard glass plain microscope slides. They're going to come in a package like this. You'll need cover slips. These are also glass, just standard cover slips for a microscope slide. Now at Aquafix, we use a micropipetter, 10 to 100 microliters. This isn't something that you need to use, and today I'll show you how to prep using just a standard transfer pipette. I have my mixed liquor sample here. I'm just going to mix this. Just gently invert. We don't want to disrupt any of those floc structures or anything. And I'm going to start just doing a standard slide prep for microscopy. So I'm going to take a little bit of sample. I'm going to put a drop right in the middle. That's all we need. I'm going to set this to the side. I'm going to grab my cover slip. Make sure I only get one. These are very thin, and so it can be easy to grab multiples. And what I'm going to do is I'm going to hold my cover slip on a slant so it covers the drop that I placed on the microscope slide. I'm just going to drop it on place, just like that. And now I have my prepped slide that I can place into my microscope, and I can look at this under the microscope. What we'll also prepare is a smear. So this is going to be what we're going to use for staining, and I'll prepare 2 of these. So I'm going to do something very similar. I'm going to take a small drop of my mixed liquor sample. I'm going to put a little drop on each, just a very small amount. And then I'm going to use the end of my transfer pipette. I'm just going to spread this around real evenly. I'm going to do the same thing here. Real even spread. You can do side to side, you can do circle patterns, it doesn't really matter. And now, once that's done, I'm going to let these air dry, and then we're going to stain these. So the first stain we're going to go through is called a gram stain. It's going to involve two different staining colors, a iodine, and a decolorization step. Now, the first step we're going to do is going to be applying gram crystal violet to the dried slide. So I'm going to use a standard transfer pipette, and I'm going to draw an amount up, and I'm going to place it on the slide and covering the slide. And now this is going to be on there for 1 minute. I'm going to use a timer. You can easily use a clock and watch that as well. I'm just going to leave that on the slide for the full 1 minute. This is allowing our filaments and microorganisms inside the flock to absorb this stain entirely. There's my timer going. I'm going to take my slide with the stain, and I'm going to rinse off that stain quickly. Now, when I do this, I want to hold it near the top of the spout because I want high flow of water, but I don't want high water pressure. If the water pressure is too high, we can wash off our stain, wash off the organisms that we have dry fixed to the slide. Since we only did a room temp dry fix and we did not do a heat fix of the slide, that's necessary. And you do not want to do a hot fixing of the slide because you'll kill your microorganisms. But after the gram stain, I'll use the grams iodine, and that's going to react to the stain, locking it into cells and filaments that are prone to maintaining that stain. And you'll kind of see what I mean a little bit later. I'll explain. So I'm going to add that on, fully covering the slide again, and then starting my 1 minute timer and letting that sit for 1 minute. Well, the general principle behind the gram stain is that we're introducing a stain, which is our gram violet, and we're introducing a counter stain, which will be our saffron and red, which we have not seen yet. I add my gram violet, which gets absorbed by all my organisms, and then I add my iodine, which is going to lock that stain into a select few organisms. I'm then going to decolorizing it, washing out the stain from everything that doesn't hold on to the gram stain, and then I'm going to counter stain with the saffron and red. What that is going to give me is organisms that stain the purple or the violet are going to be gram positive, and organisms that stain the red or the pink will be gram negative. My timer is just about up. I'm going to rinse off my slide, same way, keeping it near the top. There's more than one way to kind of do this. I like to hold the slide long ways and let the water run down the slide. Place it here. Now this next step is a little bit trickier. This is the decolorizing step. Now, you need to be a little bit careful here. I'm going to use gram decolorizer. And this is a kit that you can generally buy just about anywhere online. It'll just be the standard gram stain kit. Now, I'm going to be very careful here. Now, when I'm adding this gram decolorizer, I'm going to place it on the top of my slide. I'm going to let that decolorizer run down, and I'm watching my slide, and I'm watching the streaks of purple come off. Now, as soon as I start to see my stain no longer hanging onto purple, that's my cue that my colorizing is done, my decolorizing is done, and I need to rinse it very quickly. I can over-decolorize the stain, and that'll result in everything looking red, even if it's not supposed to. So I'm going to add a little bit to the top. I'm watching carefully, and I'm done. And it's going to happen very quick like that. It's going to be very tricky at first. It's going to take a lot of repetitions. Once you get a sense for it, it's very quite simple. And one thing to keep in mind is that I don't need the full slide to be perfect. Really, what I'm looking for is at least some portion of that slide is good, because ultimately, when I'm looking at it under the microscope, I'm looking at such a small frame of view that I'm not going to see parts that I have maybe over-decolorized or under-decolorized. Now this next and final step, I'm going to be using my Graham Safranin, my Safranin Red, and this is going to be my counter stain that I talked about before. And again, I'm going to cover this slide entirely, just like I have before. I'm going to start my timer, and that's going to stay on for 1 minute. There's my timer. In the same way I've done every other step, I'm going to rinse off my slide using my cold water. That's another thing that I had to mention. I use cold water for rinsing my slide. You don't want to use hot water. Again, that might wash off your organisms from your slide. I like to give it a few gentle kind of shakes, get those drops off. But this is my finished and stained slide. I'm going to set this aside for a bit and let it dry off. And then I'll be able to look at it under the microscope. Now, the second stain I'm going to show is going to be the Nizer stain. This is going to be very similar to the Graham stain in that it has a primary stain and then a counter stain. It's a lot less complex than the Graham stain, though. It is going to involve, again, a pre-purchased kit called the Nizer stain kit. You can typically find it in a few different spots. I'm going to use these two solutions, which are my Nizer stain solution 1A and 1B, and it has instructions on this on combining them together. Generally, you want 2 parts of A to every 1 part of B, and that's going to give you your Nizer solution 1 that I'm going to use as my primary stain. I pre-mixed this ahead of time. This can stay good mixed for a little bit, but you generally want to mix it as you use the stain because it doesn't necessarily stay good combined for a long time. In the same way that I did with the Graham stain, I'm going to coat the entire slide. Now, a little bit more in there. And then this one, I'm going to start my timer, but this is only 30 seconds. It is not the full minute that we've seen before. I'm only putting this on for 30 seconds. And at 30 seconds, I'm going to rinse this off in the same way that you've seen before. Running the water down the slide. Set it back on my rack. Then I'm going to use the Graham Stain 2. Now, you can purchase this. We prepare this in-house for our own use. It's fairly simple to make, but it's much simpler to just buy it in the full Nizer stain kit. I have a little bit that I've set aside into a centrifuge tube. Makes it a bit easier for just simple use. I'm going to cover the full slide again. And this is my counter stain. So once that's fully covered, I'm going to start my timer, and that's going to stay on for 1 whole minute. Alarm goes off. I'm going to wash the slide off as we've seen a few times already. And the Nizer stain is already complete. This one's much easier than some of the other ones that we've done. I'm going to set this aside, let it dry, and once it's dry completely, we're ready to look at it under the microscope. So when we do microscopy, why do we care about filaments? Why do they matter? Well, in and of themselves, they can cause major upsets within your system, including bulking, foaming, and pore settling. They're also strong indicators of plant health, so small shifts within the population of your filaments can indicate nutrient imbalances, potential toxicity, and even things like low DO. They give you insights into potential problems that can pop up within your system far down the line. So when we look at slides under the microscope, we are primarily looking at key morphological features with which to identify those filaments and ultimately identify what things are taking place within the system. We're going to go through those in the following slides, but those are generally going to be filament width, filament length, filament shape, whether or not a sheath is present, whether or not cells are visible within the filament, the shape of those cells, if visible, whether or not the entire filament displays motility, whether or not it's a branching filament, whether or not we can see sulfur granules within that filament. And then we're also going to look at some staining characteristics. So we're going to take advantage of certain microscopy tools like staining to help us identify filaments. So one of the first things that we look at when we're observing a filament is the filament width and length. So on its own, a filament is essentially just a chain of single cell organisms. And those bacteria, those organisms, they can have different sizes, which contribute to different sizes of filament, specifically different widths of filament. And then they have a tendency to sequence themselves in varying lengths of filament. And so what we can do under the microscope is we use the small scale bar. In the images you can see here, that's identified as a 40 micrometer scale bar. We can generally use that to track how long a filament is, how wide it is, and you can use those estimates to help you identify that filament. Other morphological features that we look at with filaments are the shape of the filament, the general structure of that filament. So filaments themselves, as they grow, they can take on different shapes, either straight, bent, curved, coiled, or irregular, just depending upon what type of filament it is. In this image, we have irregular filaments. They tend to be squiggled and sporadic throughout the sample. We also look for a sheath within the filaments. So as I mentioned before, a filament is just a chain of bacteria cells, but some filaments will develop a sheath around that chain, which protects it from environment, and then some of those bacteria with that chain can die and disappear, and the totality of the filament will still remain. So we can clearly see that sheath here in this image. And the sheath is relevant primarily because only so many filaments will have a sheath, so it's a strong identifying feature. But also these sheaths can interfere with chlorination or treatment, and they can also persist after the filament death and cause some of their own issues. Then we can really zoom in on the filament itself. As you move up in objective magnification on the filament, you can begin to identify the bacteria themselves within that filament. That in and of itself is an identifying feature, because not all filaments have visible cells, even if you get very close, a very high magnification. So whether or not the cells are visible is something to note, and that's cell visibility. If you can see those cells within the filament, you can begin to identify what sort of shape those cells take within the filament. They tend to be present as either a disc, an oval, a square, a rectangle, a barrel, or variable. But this can be a little bit tricky. It's very difficult, really, to identify the shape of a cell. For instance, when you look at rectangular cells, you might look at this filament and you might think that some of these are rectangular or even barrel shaped. But we would largely call these disc-shaped cells, which can feel a little bit surprising or misleading. That's largely to do with the orientation of the shape, and it's honestly something that you'll pick up after doing enough observations, and you'll kind of recognize these filaments and what sort of cell shapes they manifest as. The next primary things we're going to be looking at are filament motility and branching. These are really strong indicators because not all filaments show these. Only a few different filaments actually show these. So motility within filaments is whether or not the filament as a whole is moving or whether or not it's stationary. The vast majority of filaments are stationary. In fact, there's only two types of filaments that you will see moving on their own. Now, when you are looking under the microscope and you're looking for motility of filaments, it's very important to recognize the general flow under the microscope. For instance, when you're looking under a microscope, you're going to see things moving under a wet mount microscope. Things are going to be flowing in a particular direction, and those are just natural currents within your slide. You need to look for movement that's distinct from that flow. It's going to be a very particular flexing or wiggling movement that's going to tell you that that's a modal filament. Same with motility. Branching is a very particular type of morphological feature. There can be true branching and there can be false branching. And similar to motility, there's only a couple different types of filaments that actually present branching, and they have different types of branching. And so this is actually a strong observation as well that can help you identify a filament. Lastly, we're going to talk about sulfur granules and staining. Sulfur granules are mostly visible under phase contrast. It's going to be very difficult, if not impossible, to see these under light microscopy, but they exist as little tiny glowing specks within your filament, and they are very useful because only so many filaments will store sulfur, and so they're a strong indicator as well. Staining is a very useful tool to have in your tool belt for identifying stains. Primarily when you're doing identification via staining, you're going to be doing gram staining or Neisser staining. We have a short section going over staining, but it's worth remembering that it's important to remember that gram stain is going to be purple if it's gram positive and red if it's gram negative. And the Neisser stain is going to be purple if it's Neisser positive or brown if it's Neisser negative. And these indicators, these gram positive, gram negative, Neisser positive, are going to be useful in identifying your filament. Now we're finally on to filaments. In the following slides, we're going to show you different filaments. We're going to talk about their names, their morphologies, and we're going to talk about what sort of causes they have. As we move forward, you're going to recognize that some filaments have type numbers and some filaments have species names. In order for a filament to have gotten a species name, that means that the bacteria that causes that filament has been isolated and it's been speciated. Now, not all filaments, even if they're technically the same type, are really the same species. So the type system has been created as a way to group similar filaments together, and that is very useful in creating a box for us to put these different sets of traits in, but it makes it difficult to identify filaments at times. There's also some filaments where we have a slash. So for instance, S. natans / type 1701 or type 0041 / 0675. Now, we have further grouped these filaments together because they're so similar that they are often interchangeable in identification, and they cause the same sorts of issues, and they're caused by the same sorts of issues. And so they're very easy to identify together. First, Beggiatoa. Beggiatoa is one of those filaments that is going to be very easy to identify, and it is because this is one of the two filaments that I mentioned before that is a motile filament. That means it's a moving filament. It's a large, fat filament. It's thick, but not quite that long, and it moves in a sort of sweeping, wiggling pattern. If you have phase contrast, you'll also see a lot of sulfur granules within this filament. This filament almost exclusively shows up in systems that have low DO. So this is a filament that grows in an environmental condition, and that environmental condition is low DO largely, or septicity, or high sulfur present within your waste. But all of these things sort of go hand in hand. Flexibacter is our second filament that is motile. It gets its name from the way that it moves. It moves in a sort of flexing motion. It's much smaller and very skinny compared to the Beggiatoa, which we talked about previously. And so it's very easy to distinguish these two motile filaments. The Flexibacter has this very distinct dashed gram-negative pattern. You can see in the image to the left that it has this red or pink stain indicative of gram-negative, and it is a dashed pattern. It's very unique to Flexibacter. Its primary cause, similar to the Beggiatoa, but distinct, is septic or settled sludge. It tends to grow within that settled and septic sludge in your system and then swim about throughout the rest of your system. Now, it doesn't cause any issues in and of itself, but it is a strong indicator of septicity, and that septicity can be causing its own sort of problems. Helicobacter hydrosis, or H. hydrosis, is a very straight, rigid, gram-negative filament. You can't see the cells within this filament generally, and to me, I tend to remember this filament as being almost fiberglassy in the way that it lays on top of itself and extends from flock. It's thin and short, and it is indicative of a low DO in your system. When it grows to very high abundances, it can interfere with settling. You can imagine having a whole bunch of these filaments growing out and extending from flock and interfering with that flock's ability to settle into the bottom and compact well into each other. Nostocoida limicola, or N. limicola, is a fat filament. It is one of the few filaments that is Neisser positive. So we can see in the images here, we have Neisser stained images, and we have that positive purple. It is a sort of coiling filament at times, but it can also be irregular as well. It can kind of coil and loop on itself. It can also be wiggly. It almost has like a worm appearance, but obviously it isn't a worm because we're talking about sizes that are much too small to be any sort of worm. This one is mostly caused by volatile fatty acids or breakdown of fats within the system. It can be a symptom of mild septicity, and it can also occur at times during phosphorus deficiencies, although those latter two causes are less frequent than simply having a lot of fats and fatty acids within your system. Type 0092 is a smooth, Neisser positive filament. It tends to grow in those straight, longer patterns, and it is mostly caused by fats, oils, and greases within your system, as well as potentially volatile fatty acids. Now, this filament is associated with warmer temperatures, and it does cause foaming. When we see foaming events with type 0092, it tends to happen in warmer conditions. And this is largely because when it's warmer out, type 0092 outcompetes its sort of sibling filament, which is Microthrix parvicella, which we will talk about a little bit later. Type 1851 is a very thin filament, and it tends to grow in these bundles. You can see on the right a great example of that, where you have a bunch of these filaments growing side by side in this sort of bundled fashion. They also have a lot of attached growth. They're one of a couple filaments that tend to have a lot of attached growth, but almost all filaments with sheaths can have attached growth. This one is very indicative of low FDM in a system, but it's also very common. So you'll see this filament at reasonable abundances in almost all of your samples. S. natans / type 1701 is a filament that has that false branching morphology that we mentioned before. You can see this very clearly in the right-hand picture, where when you're zoomed out, it would appear that this filament branches, but as you look closer, you recognize that it's actually two distinct filaments that are kind of laid right next to each other and then diverge, as opposed to being truly a single cell which has branched into two more cells. It is a sheathed filament with sausage rod-like cells, and it is gram-negative. It's very indicative of high soluble BOD and low DO. These two issues go hand in hand. When you have a lot of soluble BOD in a system, it can cause a lot of oxygen demand, which can crash DO. Also, acetinations type 1701 tends to grow in sewer lines because it likes very low DO symptoms, and it can wash in from your sewer lines. This one can be fairly problematic and cause a lot of bulking issues if it grows into very high abundances, and it can be very difficult to get rid of because it has that thick sheath on it that when you actually deal with the filament itself, this sheath is left behind that continues to cause problems. Now, we have a group of filaments called the Thiothrix filaments, Thiothrix species specifically, and these are characterized by rectangular cells, gram-negative, and sulfur granules. And these are another one of those filaments that's largely caused by low DO, and it can cause problems similar to other low DO filaments or other larger filaments, where it's mostly going to be coming in the form of bulking and interfering with settling. Type 021N is a specific filament that falls within those Thiothrix type filaments. This one has disc-shaped cells. It's generally gram-negative, although you can see gram variability, which is actually what we see in that top left, in that left-hand image. We see that filament kind of going from red to purple and displaying gram variability there. It is a longer filament that tends to be a bit on the thicker side that gently curves. It is caused primarily by volatile fatty acids, but more so it is indicative of a nitrogen deficiency in your system. But this goes hand in hand in the same way that volatile fatty acids and soluble BOD can cause low DO. They can also cause nitrogen deficiencies, as you have a bunch of carbon entering the plant, and that throws your carbon to nitrogen ratios out and manifests as a nitrogen deficiency. Now we finally come to Microthrix parvicella, or M. parvicella. This is that sibling filament to type 0092 that I had mentioned before. This filament is very distinct compared to type 0092. It is a coiling, smooth filament. It has been described almost as spaghetti. When you're looking at it under the slide, it is a gram-positive, very strong gram-positive, so very purple filament. And this one grows in the presence of fats, oils, and greases, and it likes lower temperatures in the sense that it tends to outcompete things when temperatures get a bit colder. It is a foaming filament, and so it's going to drive foaming within your system. It is unique in that it has these Neisser-positive granules when you Neisser stain the filament. So you can see a brown filament with these purple dots within it when you do the staining. We'll talk about type 0041, 0675. The morphology on these filaments are square-shaped cells. They have attached growth. They're very similar to type 1851 in that sense. They're gram variable, but they tend to manifest as gram negative. They're very wide, and they're indicative of low F to M. Like our other low F to M filaments, they're very common within systems, even when they're not under low F to M conditions. But when they grow to extremes, they can cause bulking and other issues. Nocardia-like organisms, or NALO, are one of the more common filaments that you're going to see in your system, and they're one of the more common filaments that cause issues within wastewater plants. They are a filament that causes a lot of foaming events, and you can see that they are one of the filaments that have true branching. They're also very strongly gram positive, and when you look at them under a Neisser stain, you can also see Neisser positive granules similar to M. parvicella. Now, as a sort of final notes on filaments, they offer really great insights into process conditions within a wastewater plant. When you look at your different filaments, they can be very overwhelming at first, but it is important to remember that you are not going to be needing to identify every single filament when you're doing routine filament identification. You really are only going to see the same sorts of filaments over and over again as a wastewater operator performing routine microscopy. And it's going to be when you see a new or unique filament that you're going to have to figure out what that is. And it's also probably going to be a filament that's going to be causing issues for you. And as a final note, it's very important to pair microscopy with operational data so that you can best identify or best predict things that might be happening. For instance, if you recognize that there is a trend in lower DO numbers, and then you also see that you're getting a low DO filament increasing in population, you really know that your low DO is triggering a filament bloom, potentially, that can cause problems like bulking or foaming. And so it's important to get ahead of that, increase your DO within your system, and prevent that from being a system upset. Thank you for attending this webinar. If anything, I hope that this video has done a good job of introducing how filament identification is performed and has done a reasonably well job of giving you examples of filaments and what sort of morphologies that they have, so that when you do microscopy on your own, you can have examples of those filaments that you might be looking at under your microscope.